So, hi, my name's Michael Hansen. I currently work with the USGS at the Olympic Research Station, but I would just want to share some work that I did with the National Park Service over the past several years. This was also part of my master's thesis with Oregon State University, and it's a telemetry project on the fall and winter life history for little brown bats and California Myotis. Could I do the next slide? So there's been an increased interest in winter bat ecology with the emergence of white nose syndrome. So this is a fungal disease that's killed millions of bats, mostly on the East Coast. And it's caused by a cold adapted fungus called Pseudogymnoascus destructans. And so this fungus can only grow into the tissues of bats when their body temperature is low, when they're in periods of torpor during hibernation. And so for the first 10 years of the outbreak, this was restricted to the east side of the Rocky Mountains. And then in 2016, it jumped to Washington State. Slide. And in response, the National Park Service developed a regional white nose response strategy, but the focus of this was on bat monitoring and white nose surveillance. But the big problem is that we don't actually know where most bats in the West hibernate. And this is an issue. It means that we can't observe the animals during the time of year that they're actually susceptible to the disease. So slide. This is due to a couple pretty major regional differences in life history. So bats on the East Coast, when they go to hibernate, they tend to get together in these huge groups in caves and mines, and they can be thousands of individuals and migrating hundreds of kilometers to these communal hibernation sites, which are called hibernacula. And on the West Coast, we really haven't located very many of these. Only a handful of telemetry studies have actually tracked bats to their hibernation sites. And so this image down here is a hibernacula found in Alaska a couple of years ago. And this is really sort of representative of what we're finding for Western bats. It's like individuals or small groups in these really dispersed little crevice hibernacula across the landscape, and usually relatively close to their summer grounds. So our model for hibernation ecology is mostly based on these East Coast populations. So Western researchers now are just trying to sort of gather together like basic life history for Western bat species outside of the summer season, which brings me to my project. So slide. And it's exactly that. It's gathering baseline life history information for a couple species from the genus Myotis. And Myotis are the most susceptible of all the genera of bats to white nose syndrome. So we're looking at the types of roosts that they're using in the fall and winter, trying to document some movements, timing of seasonal shifts between different roost types, and then just compare some differences in these factors between two different species and two different populations at Mount Rainier National Park. So our two species are little brown bats and California Myotis. So little brown bats are one of the most common, widespread of all the Myotis species, but they've had huge population declines from white nose syndrome. And we actually have white nose positive groups of little brown bats at Rainier. And so we were really interested in tracking their movements, hopefully to find their hibernacula for disease monitoring. And then on the other side of the scale, we have California Myotis. And so these guys are the only Pacific Northwest Myotis species where we haven't detected the fungus that causes white nose syndrome on them in any of the disease monitoring efforts so far. And so this could be because they're also the most active of all the Pacific Northwest Myotis species in the winter. So we have acoustic recordings of California Myotis on the landscape throughout winter. They're still considered a hibernating bat species, but only a handful of telemetry studies have actually tracked them, like tracked their movements. So all we really know about their lives, well, and those studies were all in the summer. So in the winter, all we know is that they do appear to be more active than other species. And it makes sense that a disease that infects bats during hibernation might differ in a more active species, but we just don't really have a good picture of what those activity patterns are. Slide. So we had a couple of predictions. We thought that during tracking, little brown bats would probably transition from like fall habitat, which is generally in like buildings and snags, to rock or soil crevice hibernacula. And we thought that California Myotis might remain in the area where they were captured, but potentially transition to a more thermally stable roost substrate and remain intermittently active throughout the tracking period. Slide. So we trapped bats at two different locations. We had on the west side this T Woods location. That's a mixed roost of little brown bats and California Myotis. And then on the east side, we had the Ohana population, And this is a maternity colony. So this is where adult females come together to give birth to their pups. And this was our white nose positive colony. So we were really trying to find a hibernation location for these guys. Slide. So to track the bats, we applied these teeny tiny little nanotag transmitters. They're just 0.26 grams, and they're a radio transmitter, but they have a really short range because they're so small, and there's not a lot of roads in the area. So we did most of our tracking on foot, but just to supplement that, we did several flights from fixed wing aircraft, and you can see an overlay of all of those flight tracks on the slide. And next one. All in all, we telemetered 28 bats, and we located 38 different roost structures. And I just want to focus on a couple of the results that I thought were the most interesting. So we expected little brown bats to transition from fall habitat to these like rock or soil crevice hibernacula, but unfortunately, we were not able to track any little brown bats to their hibernation sites. This was kind of the primary goal of the project, so that was pretty disappointing. All of our bats did eventually disappear from their study area, and most of those were right in mid-October, which aligns really well with the onset of hibernation in studies at BC and Alaska. So it seems likely that this disappearance of the signal represents a shift from fall to winter habitat. So that could mean that the bats traveled more than the 30 kilometers that we searched from the plane. But just based on other studies, I'm guessing they went into the ground and that blocked the signal and we couldn't find them anymore. But interestingly, the bats at that western site at T Woods were detectable on the landscape for 29 days longer on average than the bats from Ohana. And so, like, we can't give a definitive reason for this difference in behavior, but it did turn out that bat was an exclusively male roost for the little brown bats, and fall is when the species mates. So it's pretty common to see males active on the landscape a little bit later, like usually a week on average, so that they overlap with the latest of the hibernating females just to maximize mating opportunities. And this difference in the timing of their signal disappearances could suggest some kind of difference in mating strategy, like potentially going between hibernacula to mate with females. But obviously I didn't observe those behaviors, really small sample size, just an interesting jumping off point for future studies. And next slide. So next slide again. For California Myotis, for these guys, we thought that they would probably stay in the area where they were captured and be detectable, and they were. We were able to track them all the way through the end of telemetry in mid-December. We also thought that they would transition to a more stable roost substrate, which they did not. Like, these pictures are bats on the outsides of buildings, really exposed roosts. We also saw them using snag roosts all the way into December, which I haven't seen documented as an overwintering habitat for them in the literature before, so that was interesting. And then next slide. We didn't see them change the types of roosts they were using, but what we did see was a difference in the amount of time that they were using each roost compared to records from the summer. So that's what this figure shows. We've got date on the x-axis, and then we have these two distinct time bins that we observed on the y-axis. So we saw bats using roosts for either 2 to 6 days or 12 to 20 days, and then all the different colors are different roost locations. So this is interesting because the 2 to 6 day period mirrors really well how California Myotis use roosts during the summer, how frequently they switch. And then that 12 to 20 day period, stationary in a single roost, overlaps really well with the average bouts of torpor for other Myotis species. So all hibernating bats do have to wake up periodically during hibernation just to like eliminate waste. But this pattern suggests that maybe California Myotis are having similar like chunks of torpor, so like short periods of hibernation to other species, but during those arousals, rather than just eliminating waste, dealing with some other metabolic requirements and going back to sleep, they might be shifting back towards something that resembles their summer behavior for a period of days and going out and foraging. So that could explain some of the differences in detection that you see between them and other Myotis species in the winter. And again, sample size of 4 is not ideal. We definitely need temperature measurements to determine exactly when bats are in torpor and, to track them later into the winter, but it's an interesting initial observation. Slide. So yeah, these are great initial insights, but there's a ton more that we need to know about bats in the fall and the winter. So I just want to quickly introduce another project that we're working on the Olympic Peninsula. So this project focuses on Myotis, and Myotis comes from the Latin for movement. And this is basically a network of stationary receivers, and they can detect the signals from tags for all of the, like, this collaborative of researchers across the world. All the tags that are deployed, then that information gets transmitted to a centralized database, as long as those tags pass within range of one of these towers. And so we put in the very first Myotis tower at Olympic National Park over this last fall, which is great because this is going to then gather information from our project, but it'll also just expand the coverage of the Myotis network as a whole. And next slide. So this is an image of that Myotis station. It's on the Glines Canyon Dam. And so this is overlooking the Elwha River, and you can see that alder cover is where the old Lake Mills reservoir bed was before the dam was removed. And so this array of antenna is then connected to a solar powered receiver, and then that transmits data via cell network back to the Myotis database. So we're able to remotely gather the data from so far just 10 individuals that we captured over the fall. In this case, it was still Myotis, but Yuma and Little Brown. And can I go to the next slide? We just have some really preliminary data here. We've got dates on the y-axis, time on the x-axis, and I've just shaded the area that represents nighttime. So each of the different colors is a different individual bat. So you can see that we already have some cool preliminary data coming in, a few different kinds of behaviors, little short bursts for commuting, foraging, and then longer term periods for roosting. And looking so a closer analysis of this, we should be able to determine sort of what they're up to during these detections. And our plans over the next couple of years are to expand with one more tower in Olympic and potentially towers in other network parks in Washington. And that's it. Slide.
Thanks, Michael.